Factors that affect enzyme activity by Ace Academia. Okay, first is temperature. Temperature affects the enzyme activity. So as the temperature rises, kinetic energy increases. And this is because temperature is the average kinetic energy of the particles. So if you have a higher kinetic energy as the temperature increases, this means they're going to be faster moving because kinetic energy is movement energy. And the faster they move, the more frequent the collisions between the enzyme and the substrate. The more frequent the collisions, the more effective collisions will happen. And the more effective collisions means that more enzyme-substrate complexes are formed. And it's these enzyme-substrate complexes that makes the reaction occur. So as the temperature increases, so does the enzyme activity, but only to a certain point. After a certain temperature, the rate of enzyme activity drops to 0. And this is because above a certain temperature, hydrogen bonds and other bonds in the active site start to break. And this makes the shape of the active site change slightly, meaning that the enzyme is no longer complementary to the substrate. And this means that it cannot fit onto it and form an enzyme-substrate complex. So then, after another high temperature, usually 60 degrees in humans, the bonds are so disrupted that the enzyme stops working completely because the active site has completely lost its shape and will not form an enzyme-substrate complex as it is completely the wrong shape and not complementary to the substrate. And at this point, we say that the enzyme is denatured. And this means it's permanently changed shape. It cannot be reversed. As I've said before, now the active site is no longer complementary to the substrate, so the enzyme activity drops to 0 because the enzymes do not work. As you can see here, this shows an enzyme and the substrate. And after the temperature, the enzyme has denatured and it's changed shape, so it no longer fits this substrate. So why are our bodies at 37 degrees if the optimum temperature of many enzymes is 40 degrees Celsius? Optimum temperature basically just means the temperature in which the enzymes work best at. So if many of the enzymes in our body have an optimum temperature of 40 degrees, why are our bodies 37 degrees? And this is because even though it may increase our metabolic rate slightly with the increase in temperature, all the advantages are undone by the fact that we would need quite a bit more energy to sustain this increased temperature, meaning we'd have to increase our food intake. Also, when we're ill, our temperatures rise. So if our temperatures internally was already 40 degrees, having to rise even more when we're ill would denature our enzymes, meaning that many of our bodily processes won't work very well or at all. For example, digestion. Next, pH. pH affects the enzyme activity. pH is the measurement of the concentration of H+ ions. And the optimum pH, the pH in which the enzymes work best at, varies for each enzyme. So generally, the pH alters the charges on the amino acids in the polypeptide chains because enzymes are proteins, meaning they're formed of amino acids in polypeptide chains. And so if the charges on the amino acids are changed or altered slightly, this disrupts the tertiary structure, which forms the shape of our enzyme and our active site, meaning the change in the shape of the active site would mean that the enzyme substrate complex would have trouble forming. Also, the change in the concentration of H+ ions, if it's big enough, may disrupt the ionic bonds between the amine and carboxyl group. The amine and carboxyl group are on each amino acid, and it's the ionic bonds between them that helps form the active site, with a few other bondings, of course. But if these ionic bonds are disrupted by the charges on the H+ ions, this can affect the shape of the active site and affect how well the enzyme substrate complex is formed, because the active site has to be complementary to the substrate. But just a key note, fluctuations of pH, so increases or decreases, in organisms are very small generally. So the changes in pH don't usually stop the rate completely, as you can see here. They just decrease it slightly. It's very rare that the change in pH is strong enough to completely stop the enzyme activity. So calculating pH is the last point. So this is the formula for calculating pH, the concentration of H+ ions. The pH is minus log 10 of the H+ ions. And so, for example, calculate the pH of a solution that has a hydrogen ion concentration of this number here. So it's very simple. You go onto your calculator and you get the log formula and you type in minus log 10 and your concentration of H+ ions, which gets you to pH of 4. So our solution has a pH of 4.